want to thank Pascal and Arvin, who disappeared, for inviting me to this meeting. So we had a few experimental talks today with Paolo and Sophie, and then my last one was also me. So there was very little theory. I hope the theorists will find it interesting and not be completely out of space. So in my lab, we study a brain region called the basal ganglia. That is subcortical. So maybe I thought what would make me the odd one out here is that I do experiments, not theory, but it turns out that it's actually because I'm not working on the cortex. So very little about the cortex, mainly in subcortical nuclei called the basal ganglia. And the basal ganglia are involved in many motor functions and are responsible for many motor dysfunctions, most known as Parkinson's disease, but also Huntington's, Tourette, and many other things are localized in basal ganglia networks that dysfunction. So so the two main processes that basically all neuroscientists work on some way or another is the integration of sensory information and production of motor output. So we can study it in pure sensory or motor neuroscience, multisensory, integrative neuroscience, learning, learning of behavioral sequences, learning of different sensory pairings, but it's always integration of sensory input and production of motor output, which is done by all levels of the nervous system. So it can be very simple reflexes like the gill reflex in dysplasia. It can be just normal reflexes to aversive sensory input, but it can also involve very sophisticated, complicated movements and very simultaneous integration of sensory information from different modalities, different parts of the body to produce complex motor output. So if we look, for example, on a violinist that is playing a concert, you can appreciate that it does almost effortlessly multisensory integration in the sense that he has to read the notes, listen to his own music, adjust the fingers according to what he hears, feel the tactile feeling of the strings and the bow, and the motor output is bilateral. Every hand is doing something else, and all of this is done on the fly. So this is an example where you have to simultaneously integrate information from different sensory modalities, different sides of the body, and produce the motor output. And as I said, the whole nervous system is involved in this kind of process, from the cortex down to the spinal cord. The region that we are interested in our lab is the basal ganglia, and the basal ganglia is made out of several subcortical nuclei. And they were mainly traditionally studied in the context of motor control. And the reason for that is the very strong motor deficits associated with the basal ganglia, mainly Parkinson's disease, which is a very prevalent disease, Huntington's, Tourette, OCD, many motor centric deficits are related to the basal ganglia, but we know that they're also involved in sensory processes. If we look at the circuit organization or the gross network organization of the basal ganglia, we can see this variant of the box and arrow structure. So what is covered here with light blue is what is defined as the basal ganglia nuclei. The main input layer is the striatum, and the striatum projects via the direct and the indirect pathway down to the output structure, which is the pallidum, which is the SNR and the GPI. These are inhibitory neurons that are tonically active, that suppress all the potential movements that we could be doing right now. So when you sit here quietly and politely listening to the talk, the neurons in your substantia nigra, pars reticulata, and your GPI are ticking on constantly at about 40 hertz, firing and inhibiting the motor centers that will suppress all the movements that you could do. Why we do a movement is because there's a temporary disinhibition. So some of the population within this box of the arrow, within the substantia nigra, they stop firing or fire less, and that helps us to release the movement that we intend. The main driving force to the striatum, what you see here are in red are inhibitory structures. So most of the basal ganglia is made out of inhibitory nuclei that inhibit each other and cause this kind of inhibition of inhibition, and the movement is released by this inhibition. So we have different pathways, and the main driving force for the basal ganglia comes from the cortex. So the many cortical regions project to the striatum, sensory, motor, prefrontal regions, so all the regions that both Paula and Sophie covered in this talk and more project to the basal ganglia. And as you can see already in this structure, many pathways that connect the cortex to the output structure of the basal ganglia, the SNR. So there are many ways that information from the cortex can reach the SNR and control behavior. The direct and the indirect pathway are the initial projections from the striatum. So for this, we have the corticostriatal pathway. This is a direct glutamatergic input to the striatum. Then the striatum projects either directly with the inhibition to the SNR or via the GP SDN to the SNR. So you have either disynaptic inhibition used via this pathway or trisynaptic disinhibition. Both of them cause or the release of movement or the stopping of movement. Then we have the hyperdirect pathway, which bypasses the striatum and is from the cortex to the subthalamic nucleus. It's an excitatory small nucleus that then excites the SNR and also excites the GPE. So we have a complication of many different pathways. So you can have either disynaptic excitation, cortex, SDN, and SNR, or trisynaptic inhibition if you go through the sign reversal of the GPE. So we have many different pathways that can enable us to bidirectionally modulate cells in the SNR, suppress or to release motion. So you have a constant rate, and you can either elevate the rate of these neurons or decrease the rate. So you have bidirectional control on the different movements that you want to produce. The way we study the basal ganglia in my lab are twofold. So if we roughly divide the experiments, we have experiments in which we work with the whole animal. We call them in vivo experiments, and we can record from individual neurons, like Paolo showed using the whole cell, the patch clamp method, where we can record both the activity of the neurons, the spikes, and the inputs, the inhibitory and excitatory inputs that they receive. So we record intracellularly, and we can see all the synaptic inputs that they receive from many other cells in the network. We can also use the in vivo recording to see how the animal responds to sensory stimulation, multi-sensor integration from the whiskers, auditory, visual, and see what happens during behavior when the animal is whisking or walking, etc. To understand the circuit and how the neurons are connected to each other, we use slices, and we do the patch clamp recordings from several electrodes from different neurons in the region, and try to understand how the different cell types or the different instruments in the orchestra talk to each other. And there are many different types of neurons in the striatum, in the globus pallidus, in the different regions. And we are interested in their microcircuit organization to understand how they are synaptically connected to each other And of course, combined with these electrophysiological methods, we also use optogenetics, chemogenetics to perturb certain pathways and certain cell populations. Okay, so today I will talk about three different projects that we had in the lab. Two already published, and one is in the way, describing the different pathways connecting the cortex and the basal ganglia output, the SNR. So we'll start with the corticostriatal pathway that targets the direct and indirect projections from striatum. So this is very old work that summarized my PhD, where I was interested in cortical networks. And one thing that was very striking in the working on pyramidal cells and how they talk to their neighbors in the microcircuit is the fact that the same pyramidal cell producing a spike train can give a completely different message to the targets based on the cell type. So here we had the parvalbumin, the somatostatin neurons, neighboring part of the cortical circuit. If we stimulate a pyramidal cell with a train of action potentials, you can see a dramatic different message given to the PV, the basket cells, the parvalbumin expressing interneurons, or somatostatin expressing interneurons, where you have a very clear facilitation during the train compared to very strong depression. So they act as high pass or low pass filters of the firing frequency of the pyramidal cells. So the same presynaptic cell can give a very different message depending on the target populations. Inspired by those experiments, I wanted to ask how the cortex talks to the striatum. So we know that in the striatum we have many different cell types. We all receive cortical input. How does the cortex talk to the different types of striatal neurons? And this was the PhD project of Yvonne Johansson, who was in my lab a few years ago. And then we studied different presynaptic regions, either from somatosensory cortex, primary motor on the other side, the contralateral one, and thalamic inputs, and recorded the responses of 5 different types of neurons.
So, Gilly,
yes,
in that slide,
yeah.
So the frequency of stimulation, was it different then?
Here this is an example of a 40 hertz stimulation. This is of 20 hertz. But the synapse is depressing or facilitating.
So even if you use the same frequency,
yeah. Of course, okay. yeah. The synapse, the synaptic mechanism is very different. The calcium dynamics of the boton is
Thank you.
Different. So it's the axonal botons you're talking about, and they have a different dynamics, or is it the synaptic and postsynaptic? Because that's more
the postsynaptic identity.
Yeah.
Determines also the type of synapse that will be formed from the presynaptic side.
Sure. Okay, but it's not a dynamics that's inherent in the axon.
In the boton, in the specific boton.
Axonal boton.
Yes.
Okay.
Yes. Okay, so in these experiments, we expressed channelrhodopsin in this case in cells in S1, primary sensory cortex, and recorded from identified neurons in the striatum and stimulated with light the axons that come to the striatum. And the idea is to record simultaneously from different types of neurons, either projection neurons, the MSNs in the striatum, or parvalbumin neurons, or somatostatin neurons, or cholinergic interneurons, and see how the cortex talks to the different elements of the striatal circuit. And this is how the experiment looks like. We can identify the different types of neurons in the striatum just using their electrophysiological properties or a fluorescent marker, stimulate the terminals with blue light. There is a synaptic activation, and we can simultaneously compare neurons in the same place of different types, how they, what is the size and what are the properties of the response, And we could see that there were very dramatic differences between the cell types. We repeated these experiments from motor cortex, from thalamus, from the other contralateral hemisphere, and we could get a map that reflects both the presynaptic region and the postsynaptic cell type. And what stood out is that these parvalbumin cells, the fast spiking interneurons, had the strongest responses, excitatory responses, and they provide feed-forward inhibition to the MSN. So from all regions that we tested, they were the highest 100% connectivity, and always highest synaptic amplitude that they received from the cortical and the thalamic regions. The situation was different for the low threshold cells that did not receive anything from the contralateral from the thalamic side. The cholinergic interneurons didn't receive anything from the contralateral side. So there were certain pockets of selectivity in the way that the cortex talks to the striatum. Also imbalance between cells that belong to the direct or the indirect pathway, dependent on the identity of the presynaptic region. So the overall strength, when we talk about the way that the cortex talks to the striatum, depends on the identity of the postsynaptic cell type and which cortical presynaptic region we are talking about, and of course the thalamus has its own particularities. And from all of the neurons in the striatal circuitry, the ones that constantly received, the population that received the strongest input was the fast spiking interneurons. In parallel to these slice experiments, we were also interested in the sensory properties of the striatum, and for that, a postdoc in the lab, Ramon Reg, he embarked on a very ambitious, and that was the first time that anyone did these experiments, which were to try to record intracellularly using whole cell in vivo recordings from the deep striatal neurons. And he managed, he first of all wanted to see how they respond to whisker stimulation, so he injected the dye in S1 in the primary sensory somatosensory cortex in the barrel field where it integrates the whisker responses, and traced the axons going to the striatum, put the pipette in the intact animal there, and recorded the activity of the neurons in the anesthetized animals first. So when you do the normal ketamine psilocin anesthetic, I have these typical slow wave that are typical also of slow wave sleep, very sub hertz up and down states, and this is how an MSN, a normal medium spiny neuron in the striatum looks like. So they are the majority of the neurons. They are usually very, very quiet. They are hyperpolarized. They will only fire if they get strong enough input. So they are very quiet. If you go with an extracellular recording electrode, you will miss hundreds and hundreds until you see some activity. So there are many silent ones that will be completely sub-threshold. And here we can record the activity. We can record the synaptic input that they receive during the up states and the down states. If we inject a bit of current, we can elevate the membrane potential and see the spikings riding on the up states. In about half of the cases, we could also find the neuron and localize exactly where it is lying in the striate. So this is the spontaneous activity. And as you can see, this activity is very strongly locked to the cortical activity. So what we can see here is the sub-threshold oscillations in the neuron, in the MSN. What we see above here is the field, local field potential in somatosensory cortex. You can see that it is very, very locked, and the main driving force for these neurons is the cortical input we studied in the other project I showed. So the cortical input is the main driving force for this spontaneous activity and also for evoked activity. So what we can see here is responses to contralateral, ipsilateral, or bilateral whisker stimulation and using different types of silencing via TTX, we could show that the main source for the sensory responses in striatal neurons is the ipsilateral cortex. So the direct projection from the same from the somatosensory cortex in the ipsilateral hemisphere. All the other connections are cortico-cortical, and eventually what drives the striatum is the ipsilateral connection from S1 to the striatum. And here we can see what happens to both the LFP and the synaptic responses when we apply TTX locally in the somatosensory cortex. Okay, the next question we had about this sensory stimulation is what happens when we marry together the sensory and motor activity. So we can give the sensory stimulation, the whisker stimulation with an air puff, and we get a response. But this was with anesthetized mice. What happens with mice that are awake? Sometimes they whisk, they move the whisker spontaneously. Sometimes they're just they stop whisking. They whisk, they stop whisking. What happens to the sensory response when the animal is whisking and when the animal is quiet? To do that, we did these experiments in awake animals. We can record optically the whisking. We can see the movement of the whisker. We can record what happens to the striatum, and we provide air puffs to the whisker at different at random times. Sometimes it will be when the animal is whisking, sometimes it will be quiet. And in order to distinguish neurons that belong to the direct or indirect pathway, we express channelrhodopsin and use this device called the optopatcher, where we can combine the in vivo patching with a fiber optic that is inserted in the pipette, can stimulate and give us an immediate classification whether we are in D1 or D2 neuron. D1 being the direct pathway neurons, D2 being the indirect pathway neurons, and here we can express selectively channelrhodopsin in one population and not the other. So we can directly see whether it is this neuron or that neuron. So using the optopatcher, we could classify the neurons. And this is how a normal trace of the experiment looks like. We can see the whisking epoch with the diode. We can see the local field potential and the subthreshold activity of neurons in the striatum. And to cut the long story short, what we saw is that there is an attenuation of the sensory response when the animal is whisking. So different from what Paulo mentioned about the visual cortex, where it's the opposite. When the animal is walking, you have an enhancement of visual responses. Here, when the animal is whisking, there is a decrease in the sensory response to unexpected stimulus. And we can see it here, the amplitude of the response, both in direct pathway and indirect pathway, in a sense, is decreased when the animal is whisking. So what was the source of this decrease? So the responses that are attenuated, even in the already at the cortical level, it happens as well. So when the animal is not whisking, the response, the local field potential is higher than when the animal is whisking. So we'll do the same experiment, stimulate the whiskers and record the LFP and S1. The LFP and S1 is reduced when the animal is whisking. So already at the cortical level that then is inherited by the striatum, there is a reduction in the sensory response because of the movement. Okay, the next pathway, hyperdirect pathway. That's the shortcut from cortex to the STN and to the GPI SNR. So this cortex was this pathway was studied by what was studied extensively, and the name was coined by the lab of Atsushi Nambo in Japan. And the main thing that they saw when they stimulate cortex and record downstream in the GPI or SNR is this triphasic response. So you have the first excitation via the hard hyperdirect pathway, then you have the inhibition because of the direct pathway that inhibits directly the SNR, and then the indirect pathway kicks in and gives an increase of activity because of the disinhibition. So you have these triphasic responses, and we were interested to see, first of all, if this is happening also in sensory stimulation, not just electrical cortical stimulation, and what happens if we play with the different components of the circuit. So this work was done by Maya Ketsev. Again, we provide sensory input. She did even deeper whole cell recordings in the globus pallidus. And you see that when you stimulate with the whisker stimulation, we get this typical triphasic responses in different cell types in the GPE. They all responded to this input, and then Maya wanted to test what is the role of the STN in all this pathway. So she expressed an inhibitory opsin in the STN cells. So when we shine the blue light, instead of exciting the STN, we hyperpolarize the STN, basically remove it from the pathway. And what she could show is that the initial excitation, as well as the third the third component, were both diminished when she inhibited the STN. And you can see here that the response to baseline or baseline with bilateral stimulation or bilateral stimulation with the light. See that the whole response is distorted. A reduction in the first response and reduction in the rebound excitation. So the STN is involved in shaping this triphasic response, and reduction of the activity can affect not only motor processes like a stop signal, but also sensory responses within the basal ganglia. Okay, so all of this work was published, and then I will tell you about a new paper that just now recently got accepted, and that was inspired by this paper. So a couple of years ago, we saw this paper by Sylvia Arbour describing cells in the motor cortex belonging to the PT type of pyramidal cells that project ipsilaterally and just very deeply project to the contralateral medulla. So they analyzed the different other targets that receive input from these pyramidal cells. And we were surprised to see quite a strong innervation of the substantia nigra pars reticulata. So basically, an anatomical basis for a projection from the cortex directly to the SNR, and it had different topographical organization that reflected the type of the cortical origin of the pathway. So a PhD student in the lab, Scott, took it upon himself to characterize this pathway. And what we did at first was just the same type of experiment, expressing channelrhodopsin in motor cortex. M1 is primary motor cortex, recording from cells in the substantia nigra and seeing the responses. So you can see here already anatomically, you can see a lot of axons coming from the cortex to the SNR. And these are two neighboring cells that he patched. And indeed, when we stimulate with blue light the terminals coming from the cortex, we got strong synaptic responses. Not in all cells. There were cells that did not respond at all. There were cells that respond very strongly. but a bit more than half of the neurons responded directly to cortical input. The responses were excitatory. They could be blocked by NVQX, and we could show that they are monosynaptically using this cocktail of TTX and 4AP. It basically isolates out or filters out all polysynaptic innervation and just shows reveals that the pathway is monosynaptic. So the pathway is monosynaptic from cortex to the SNR.
So
Yes.
this is in vivo or slice?
No, slice. Yes. And the same, we then did injections in M2. So secondary motor cortex, again, got the same responses and characterized the neurons in the SNR that receive input from M1 and M2, and they were slightly different. They were located in different places in the SNR, as I will show you, and also had different levels of excitability. So the neurons receiving from N1 had a higher spontaneous level of firing compared to the ones receiving from M2, and they were more excitable. Now, we did some voltage clamp experiments to characterize the nature of the synaptic input. So we used a slightly different cocktail in the pipette, characterized the dynamics of this pathway. So they were both depressing. There was no difference between the M1 and M2. Both of them mediated by AMPA receptors and a very small, almost negligible NMDA component. So that's just basic synaptic characterization. So monosynaptic NVQX sensitive, so AMPA mediated, direct connection from cortex to the SNR. We wanted to see what is the impact of this pathway on the neurons. So when we stimulate this pathway with the trains of action potential, so activate it for a while, you can see elevation in the firing rate, both when stimulating M1 and M2. So when we stimulate the pathway, we get excitation as expected of the SNR cells, and they fire at higher frequency. Topographical organization. So we did some anatomical analysis of the pathways from both M1 and M2 to the SNR, and we could see that even though there are some overlapping regions, they had their hotspots of innervation were quite separate, with the M2 more ventromedial and the M1 projections a bit more dorsolateral. When we looked at so these are the axonal projections. Then we used a Korean flip approach to distinguish between M1 and M2 recipient neurons in the SNR, and there was very tiny overlap between those that received input from M1 or M2. So very few neurons received input from both cortical areas, and they had also a bit similar topographical organization that reflected the axonal projections of the corticonigral axons. How selective is this pathway? We did some injections in S1, like we showed before, very strong corticostriatal innervation, but nothing to the SNR. So at least in the three regions that we tested, S1, M1, and M2, only the motor regions projected to SNR, whereas the somatosensory did not talk to the SNR. So no optical responses, and no evoked synaptic responses. Within the SNR, there are also dopaminergic cells to distinguish between dopaminergic and GABAergic. And indeed, the dopaminergic cells receive only a fraction of the input that the GABAergic cells receive. So it is selective both in terms of the presynaptic regions and the postsynaptic cell type. Lastly, we wanted to see how the two pathways could affect the behavior of the animal. So they project to different parts of the SNR, and the SNR and the basal ganglia in general has some topographical organization, whether it is to promote movement, locomotion, or paw movement or grooming or different orofacial functions compared to whole trunk movements. So the approach we used here is to express dreads in the postsynaptic population within the SNR, so we could express the chemogenetic inhibitor either in cells receiving input from M1 or M2 and see how it affects the behavior. The ones that received M1, once we injected CNO that would activate the inhibition, moved less. So if we put the animal just in an arena for 15 minutes and see the overall locomotion, they reduced their movement. In contrast, the ones that received input from M2, when we did the same experiment, injected the CNO, they moved more. So they had a differential effect on locomotion, whether we study the M1 or the M2 projecting population. And here's all the data. So the kinetic parameters showed a differential modulation of locomotion, depending on whether it is M1 and M2. We did a bit more detailed breakdown of the behavior of the mice, not only if they are moving or not, but if they are rearing or grooming or doing different kind of fine movement from what we could do in the videos using deep lab cut and analysis of the behavior. And this is just an example of two mice where we can analyze the behavior, where the animal is going, how it turns to the right, to the left, what the paws are doing, basically moving the Cartesian system from the allocentric to the egocentric point of view, that we can analyze the different coordinates that were tracked markerless using the deep lab cut. Okay, so this is the way we treated the different mice, and so differences in the paw, mainly the front paws compared to the locomotion, and this is still work in progress. So what we know for sure is that it has a differential impact on locomotion. The animals that locomote more are grooming less, and vice versa. So there's different lines of the impact of the motor cortex of the SNR, depending on which region in the motor cortex. And we are trying to train the animals to do a very simple task, try to interfere with these pathways during the reach and grasp task. So we have this thing that there's a little droplet, and the mouse learns to reach, grab the droplet, and drink the water. Some of them learn the task very quickly. Some of them take some more time, but this is now work in progress that Scott is doing. The idea is to get a very stereotypic movement, sensory response. That they hear the sound, they know that the drop is coming, they reach, they grasp, and they get the reward. And then to see how this particular pathway is involved in this behavior.
Sorry, Gilad?
Yes?
I didn't really get the conform construct, because isn't that the construct that in order to be expressed needs both the Cre and the feed?
Yes. So we know it's the inhibitory neurons, the PV-Cre. So we use the PV-Cre mouse. Mice. We have PV-Cre mice, and we have the anterograde transsynaptic from the cortex in. So only the neurons that will express both Cre and FLIP will be inhibited by the dread. And now so for this learn task, this is what we plan to do in the future, is to see the different inputs to the SNR that underlie the behavior at different phases of this stereotypic behavior. So to summarize, we have the direct pathway, cortical input to the striatum, depending strongly on the type of the cortical region and the cell type within the striatum. Hyperdirect pathway causes this triphasic response to cortical stimulation, And the new pathway that Scott characterized is the corticonigral pathway that basically bypasses the whole disinhibitory structure of the basal ganglia directly from motor cortex to SNR. It's monosynaptic, AMPA-mediated, has selectivity in terms of the cortical regions and the cell types in the SNR, and seems to have very differentiated functional responsibilities. At least that's what we saw comparing M1 and M2. And that's the story. I want to thank the people who did the work, especially Scott, Patrick, Maya, Roberto, and Ramon, the data I showed, and Yvonne Johansson, and the funding agencies. Thank you.
You mentioned the topography within the strait and how also there's a certain level of organization. My thought is, within the SM, is it are these motor cortex projection and secondary motor cortex projection meeting up with their already, straight and filtered input? Can you make that pretty?
Yes.
Yeah.
So of course, if we just stimulate the motor cortex with a brief simultaneous optogenetic stimulation, we will get the immediate response in those neurons, like the initial polarization, and then we'll kick in all the filtered. Then you get this kind of signature of multiphasic responses because of all the cancellations and the delays.
Okay.
But in real life, you will have these things ongoing, and of course, excitatory input falls on neurons that already integrated some striatal input. Question is, well, what is the role of this shortcut? Why do you need the shortcut if you have so many multiple ones? And one can only speculate. It's hand waving. But I think for extreme cases when you need a really quick stop of a movement, for some reason, an alert, or you are distracted by something, if a predator comes and the mouse needs to run, need to have a very immediate stop of the movement. It doesn't care anymore how fast it's going, and if it's moving to the right or to the left, it has to stop the movement and do something else. Rapid switching of behavior, that could be a role. This role was at least assigned previously to the hyperdirect-pathway, which is also kind of a shortcut. So this is a shortcut of the shortcut, even a quicker route directly from the cortex to the motor control part of the striatum. So yes, I think in ongoing behavior, the cortical neurons will fire. They will send their synapses. They will mix with the inhibitory input that the SNR receives, or gradual shaping. I think in extreme cases where you have to switch rapidly between motor programs, this could be a useful tactic.
Yeah, this is also about the M1 and M2. So when you artificially stimulate the M1, then you have a very short latency down to the subthalamic nucleus there. Now, when the mouse is actually moving, does use the same pathway. So my reasoning is this: now, if you interfere with something that the mouse already movement already has done, then it needs to be inhibited, right? As we've seen, as the movement continues.
Yeah.
So, but the M2 stimulation, obviously, that's separate from it. But this is when the mouse is planning to do a movement, and therefore it's happily rotates.
Yeah, I think
explore.
No, I think that this homology between species as M2 as just the planning, the movement planning region, like I think the SMA or in monkeys and humans, it's not exactly the same. So in the rodent literature, at least the areas that we call M1 and M2 are just responsible for different types of movement. So one is a bit more fine movement in the front paws and the orofacial regions, and the M2 is more responsible for locomotion.
So are you saying that okay, because in primates, if I remember correctly, also the pre-motor is a parallel to the M1.
Yes, so I think there is a bit of a mismatch in the nomenclature.
Are you saying also that in awake moving mice, M2 is sufficient to elicit movement? So when you look at the cortex, only M2 neurons are active.
No.
That's not what you say.
No. I don't think it's such a black and white thing. And I think put an electrode in every place in the motor cortex or the stress, and you'll get activity. So clearly, these optogenetic manipulations are very, very artificial. It's not what is happening in real life, but it is a very good tool to dissect the pathways.
So at the beginning, you showed that different inputs onto different cell types have different strengths. So you have So I was wondering, what about plasticity properties, both short-term and long-term? Do they depend on input and/or cell type?
Yes. So we didn't do a lot of these long-term plasticity. So we worked a bit with the short-term plasticity, depression, facilitation, but there's a large body of work studying corticostriatal plasticity, and what they see is that the most plastic synapses and the most susceptible synapses in the corticostriatal pathways are the ones that go from onto the medium spiny neurons, so the SPNs, because most of the synapses there are formed on the dendritic spines, which are very, very malleable. So depending on the activity of dopamine and acetylcholine and the pre and post, there are many, many different ways to induce plasticity, but there you can really change the synaptic strength. All the classical LTP with different types of pairings show dramatic effects, especially on the D1 and D2 in a sense, and to a much smaller extent on the internodes. We didn't study that in our paper, but they are by far the most plastic. Yeah.
So very intriguing observation. These ones on the hyperdirect path, on the differential. I don't know if it can be a little contribution to the reflection, but since we are talking about the role of M2, I also think, if I'm not wrong, that even in primate, it's not easy to evoke movement with microstimulation in M2.
Even in humans,
it's not
the most frequent is stop of movement.
Exactly.
This is why I was thinking this was the switch. Just as a little. And then the second, I'm really also intrigued by this reduction of the sensory response when the animal is actively whisking.
Yeah.
And then I was thinking, maybe you touched this, since obviously some MSN must fire, right? Because the animal is whisking. Yeah. Could it be, but it's a little cluster, could it be that when you see the reduction, you are actually observing the consequence of the lateral inhibition from those that are firing because the animal is whisking?
Yes.
I'm just...
that's exactly that. I didn't go deep into that paper, but basically we showed two mechanisms that were suspect for why the whisker response will be reduced by movement. So in the cortical literature, there were experiments showing that when, like mentioned the Colciani paper, that visual responses are enhanced when the animal is moving. But the same experiment, if you record from cortical, from auditory cortical cells in A1, the responsiveness to auditory stimulus is reduced when the animal is moving.
Yeah, and then moreover, it seems to be dependent on day and night.
But you have a reduction. So in the whisker system, you have a reduction. So when the animal is whisking in the cortical in S1, the responses to whisker stimulation is reduced, and that is then inherited by the striatum. But even in the striatum, what I didn't show here is that during the stimulation, we could measure the input resistance to the, just give a continuous up and down current injection, and you can measure the constant resistance of the cell. We could show that when the animal was whisking, there was a reduction of the cell because it's bombarded by synaptic input from the cortex and from the striatum. So the cell is more conductive. And then the same input was resulted in a smaller voltage deflection. Okay, so there's also a striatal mechanism for why there is a reduction in the sensory.
Yeah, but
because the cell is more conductive.
But you agree that some MSN must actually instead fire.
Yeah, because
drive the whisking, right?
Yes.
Okay.
But they also have collaterals within the striatum,
Yes,
and
exactly.
feed-forward inhibition and many other things. Okay.
Okay, that sounds